This is, of course, an area that has not started now. This is a very famous review by my very good friend Andy Miller, published in 1991. I was 2 years old at that time. Andy was 10. He's always been really a premature scientist. But what I think, I mean, I always show this slide because it is actually the paper that has driven me to this field so many years ago, and they got really passionate. But at that time, Andy reviewed the literature of the many papers that have been published on depression and the immune system in the 80s. So this is really a field that has been developing over the years. Now, one thing I want to quickly to mention is that we have found that this is a field that resonates incredibly with the public and the patient's community. We have a digital magazine at King's College London that I lead called Inspire the Mind, and is directed to the public for kind of providing evidence-based scientific information. And we always have really good success in hit and reading of the many articles that we publish when you talk about inflammation in the context of mental health. And I think this is because of the notion that the immune system obviously is so much part of our body, that especially for the public and the patient community, the notion that you have something going on in your body that is measurable and is abnormal kind of validates the experience of illness and kind of really bridge together mental health and physical health, and is profoundly destigmatizing. So I think on top of everything that is kind of relevant from a clinical point of view, I think that this notion of this field, these two of the immune system and psychiatry getting together is also really important for bringing psychiatry and mental health in the context of what we all want, which is kind of the context of general health. Now, I'm not going to kind of summarize 20 years of research that many people have been doing in how the immune system is involved in the pathogenesis of depression. And yesterday mentioned the core mechanism that could be involved. But I think it is important to highlight that there is bidirectional communication. And it doesn't really matter where you start. Then it kind of can create a vicious circle that puts the higher level of inflammation having an effect on the brain, or the effect on the brain having a high level of inflammation. So, for example, you could start by stress, early childhood trauma or adult life stress, so that activates the brain response to stress and the body response to stress. And the body response to stress includes an activation of the immune system, even in the absence of infection. That's possibly one of the most important kind of development in neuroscience of the last few years, understanding that immune system gets activated just as part of the stress response. And then, of course, the immune system activation affects the brain through very specific molecular mechanism and very targeting specific circuits that have to do with motivation, reward, energy, fatigue, and so on. So there's a lot of information on the causal mechanisms and how this could be started either from the immune system or from the brain and the creating that. So I'm going to kind of, again, tap into Andy's work by showing, starting with showing the example of success, and because also they kind of driven some of the work that we have been doing. And Andy will talk about this, the famous trial with infliximab in treatment-resistant depression, where the overall results were negative, but as soon as Andy start stratifying the patient based on the level of CRP, C-reactive protein, so that kind of, as you all know, pro-inflammatory biomarkers, kind of synthesizing, if you like, immune activation. As soon as you kind of start stratifying patient for higher and higher level of C-reactive protein, the improvement between the patient on infliximab and placebo becomes more evident. So that's the notion of personalized medicine or psychiatry, so that if you select on a biomarker that is relevant to the mechanism, then you have an effect on patient in which that mechanism seems to be particularly important. Now, of course, we've done exactly the same things with minocycline a few years later. This is a paper that is a study that has been led by Valeria Mondelli, myself, and also many people in many of the team in the KCL team in the audience, where we gave minocycline, which is a broad spectrum anti-inflammatory medication, possibly brain penetrant, at least from animal data, to a group of treatment-resistant depressed patients on top of the normal antidepressant they were still taking. And we found basically exactly what Andy found. So it is a small study. It's a small study in sample size and a short study in terms of duration, but we found a very strong effect. This is the Hamilton improvement between week 0 and week 4. And so the higher the column, the more the improvement. And as you can see, the only group that really shows an improvement is the first group on the left, what is labeled CRP plus M. So this is the group of people that has CRP above 3 milligram per liter and received the minocycline. The group with the CRP above 3 milligram per liters on placebo don't improve, but most interesting, the group with CRP less than 3 milligrams, so the non-inflamed also don't improve. And of course, the non-inflamed, non-placebo, non-improved. So that kind of almost makes it, too good to be true. Okay, so there is a group of depressed patients that got a CRP above 3 milligrams. We call them inflammatory depression, and we give them minocycline or a similar antidepressant, a similar anti-inflammatory. And then, of course, that's what we've been aiming to do. Not all the studies have done it, and just mentioned this yesterday, and that there have been at least 2 main negative studies using celecoxib or minocycline in non-selected patient or in patient which the kind of average inflammation level was quite low, and so much so that then this has kind of pushed this paper that Andy has published a couple of years ago about thinking about really selecting patients based on biomarkers before we go for a clinical trial. The problem is that I want to kind of, after these 5 minutes of introduction, what seems again maybe too good to be true of a picture, I want to then just put some questions, then some possible answer. Or who are these people with CRP above 3 milligrams, and why should we target only them as opposed to other groups? And let's skip this for the sake of time. Sorry. All right, so who are these people with CRP above 3 milligrams? We published about 3 years ago in the American Journal, the first and still the largest studies on depression and inflammation from looking at the UK Biobank. And I'm going to walk you through key things in the table. There are lots of details, but not all of them are important. The first things I wanted to show you is that if you look at the comparison between controls and depression case, there is a higher level of CRP, 2.44 compared to 2.11 in controls, but the difference is quite small. And you probably would expect this because the controls, this is the UK Biobank, as you all know, is a community sample. So it's the dirtiest possible, the kind of more real life, yet the dirtiest possible samples to analyze from that. The control subjects are controlled only because they are not depressed, but they have also some medical and physical illness, and of course they have all kind of background and lifestyle background possible. The depressed patient, vice versa, were mostly community depressed patient. It is a lifetime diagnosis. So at the time the blood was taken, the people might have not been depressed at the time. They could have been depressed before, or they may even be depressed after, because of course the depression assessment was done a few years later after the blood was taken. So notwithstanding kind of minimizing the possibility of finding a difference, there is still a small difference. But perhaps this is the most kind of challenging issue, that of course there is no separate group of people with depression and CRP above 3 milligram liters. Is a simple shift to the right of a biomarker. So if you look at the people in the distribution 0 to 1 milligram liter, so these are the least depressed, the least inflamed or the non-inflamed individuals. Of course, more controls than depressed patients are in this group, but there are quite a lot of depressed patients in this group. In the group 1 to 3, so at what we can call the beginning of an inflammatory signal, then the proportion of patients and controls is very similar. As soon as you go above 3 milligrams per liters, of course, more depressed patients than controls are in that group. You still have controls with CRP above 3, and that remains the same as the higher you go with CRP. So there's something about 3 milligram liters, this clinical threshold that of course has been made famous, if you like, by the fact that it is associated with higher risk for kind of cardiovascular and metabolic disorders. There is something about the 3 milligram that justifies that this is when more depressed patients than controls are above this threshold, but still is not, it's not like there's nothing here and then all of a sudden depressed patients become inflamed and no controls. And even the proportion of this, if you like, is relatively small in the big population. So in the UK Biobank, which, as I said, is possible, most confounding kind of a sample possible, around 21% of depressed participants have CRP above 3, compared to 17% of the healthy subject. I will show in a second that the data in treatment-resistant depression is different. The more you select for severe forms of depression, the more disproportionate people with CRP above 3 increase. But is this just simply a kind of stronger shift to the right of a biomarker that is normally distributed, or there is something special? And that's what, I guess, the question that I'm trying to address, or at least to put some possible answers to these questions. Now, of course, the other important things that, as we all know, the higher CRP in the UK Biobank, as in basically all studies, is also associated with a lot of variables that are themselves important for depression. So older age drives higher CRP, female sex drives higher CRP, higher BMI, smoking, history of early trauma in life, low socioeconomic status, presence of physical health problem. So all of the risk factors for depression are also risk factor for inflammation. So the kind of question we wanted to address in the UK Biobank is also how much of the increased inflammation is due to these confounding factors, or is there something core, biologically core of inflammation in the context of depression? And again, as many of you are kind of familiar with this concept, both these theories are true at the same time. So in other words, a lot of the inflammation in depression is driven by kind of lifestyle and physical factors, but there's an element that is part of the biology. So let me walk you quickly through this slide. These are kind of various models with different level of adjusting for confounders. As you move from left to right, you adjust for age and sex, and also smoking and BMI. As you go from top to bottom, you adjust for what you have the unadjusted models, and then adjusted for childhood trauma, then for trauma and socioeconomic scores, and trauma, socioeconomic score, and physical health. And I guess there's two really key points to identify here. This is the least adjusted model, if you like, highly significant association between depression and inflammation. The beta value is positive. And this, the one on the bottom right, is the more adjusted, so the most conservative model possible. And you still find, although reduced association, is still significant association. So the way we've interpreted this data is as saying, yes, a lot of the inflammation in the context of depression is driven by lifestyle or physical health, or I wouldn't call them confounders. They are part of the pathogenesis of depression. So they are as important as all the other factors. They're not confounders. They are on the causal mechanisms. But there is also a core element of inflammation that doesn't disappear after you adjust for everything you can adjust for. And I think this is really the inflammation driven by the activation of the arousal and stress mechanism that I mentioned before. So it's part of the biology of depression. Now, as I mentioned before, the proportion of people with CRP above 3 milligrams tends to increase as you move from a kind of community sample to more clinically selected samples. And these are the data from the BioDep study, a large UK-based consortium on depression and inflammation. And if you look at the difference between healthy volunteers, treatment-responsive depressed patient, and treatment-resistant depressed patient, you can see that the proportion of CRP in the treatment-resistant depressed patient now comes to around 30%, while the proportion of the treatment-responsive is around 22%. So it's very similar to the UK Biobank. So it still shows that people who are depressed, people who have been depressed, who have a history of depression, they're well when you take the blood, they still have a higher level of inflammation proportionally compared to healthy controls. You will notice here also that there are, of course, people with CRP above 3 in the controls. The proportion is more in the UK Biobank because these are, again, as we all know from the clinical studies, you always end up recruiting super controls that are slightly healthier than the general population. Okay, so we know there is something about those with CRP above 3 milligrams per liter. We know that they're responding to anti-inflammatory in the way other depressed people don't. And we're showing it with infliximab, we're showing it with minocycline. So what is there something? But we also know that there's not like a separate identifiable group. These are just people that are above a threshold of a biomarker normally distributed, the shift to the right in depression. So what could be the reason underpinning this. What we have been doing with Luca, who's in the audience, we've been doing quite a lot of molecular studies in this group to try to understand the mechanism underpinning this. And especially to try to understand whether it is just like a dose response effect, and at some point you kind of, you draw a line and you say, okay, above this is relevant, below this is not, or is there something that changed biologically between as you move from zero inflammation, so CRP less than 1 to 1 to 3 and above 3. So we've been doing two sets of investigation. First of all, we looked at candidate gene expression. So this is mRNA, whole blood mRNA data. We started with candidate gene expression. This is again part of the BioDep study that I mentioned before. And look at exactly kind of tested this hypothesis by comparing these three groups of people. So the depressed patient with CRP below 1, the depressed patient with CRP between 1 and 3, and the CRP above 3. So that's the group. The CRP above 3 is the one that are supposed to respond to anti-inflammatory. Of course, we don't know in this sample because we didn't do a clinical trial in these samples, and the controls. And we look at a variety of candidate genes, most of them related to either pro-inflammatory or anti-inflammatory signal. And I'm not going to go gene by gene in this slide, but you can probably eyeball it and all notice one thing. So the three shades of gray, the three shades of green represent the different group of depressed people. So the light, as you would expect, the lighter is CRP less than one. The kind of intermediate green is 1 to 3, and the dark green is above 3. So and the yellow is the controls. And you can see that across virtually all of the genes that we've analyzed, some of them pro-inflammatory, some are anti-inflammatory, so there might be a reverse direction, but the effect is the same. There's virtually no difference between the three CRP groups. You can see most of the time there are 2 or 3 asterisks indicating that all depressed patients, irrespective of the CRP level, seems to have, at a molecular level, some kind of immune activation. So much so that, and I'll show you in a second, even the depressed people with CRP less than 1, yet they have activation at a molecular level that is, let me show the data now. So this is a comparison, a direct comparison of people with CRP less than 1 milligram per liter versus the control with less than 1 milligram per liter. So all of the kind of non-inflammatory people together. And yet you can see that across all or most of the pro-inflammatory, of the inflammatory or anti-inflammatory genes, there is a difference between the depressed patient in green and the controls in yellow. So, but this is not, of course, because these people are not inflamed in the classical way. In fact, if you measure the classical biomarker of inflammation, so the serum pro-inflammatory cytokines or the cellular subpopulation, you find that the higher level of inflammation measured in kind of through these classic biomarkers that is proportional to the level of CRP. So in other words, the CRP does reflect clinically relevant information when you look at the a measurement of downstream of the cellular output. But if you look at the molecular levels, they all seem the same. So there's something that translates genetic or epigenetic or kind of early life predisposition to an inflammatory phenotype that is present at the molecular levels in all depressed patients. In some of them that get transformed into clinical inflammation at a protein or cellular levels, the peripheral level that then is associated with the phenotype of responsive to depression. And I think responsive to anti-inflammatory. And I think until we really dissect what is the particular specificity or molecular specificity of these groups, we will not be able to understand exactly what's the best treatment. Now, we have done a few more steps to address this, and this on the last couple of slides. These are unpublished data using mRNA sequencing in this same subpopulation. The paper is under review. If the referees are sitting in the audience, I would like to thank them for their amazing comments, or if they are online. We've done everything you asked us to do. Please be compassionate. This is a really long, we've made a lot of effort to do this. So these are the results from mRNA sequencing. Again, Luca's work. And the key column, again, I'm going to walk you through because I can see there's lots of data and written very small, but the key two columns are the one in the two in the middle. So the kind of second and third from the left. These are the column of the comparison of CRP, of depressed patient with CRP 1 to 3, so at the beginning of inflammation, and then next to it, the comparison of the patient with CRP above 3 milligram, both of them versus control. So you could imagine that like a simple dose response curve, as in, you know, the same pathway being activated in both groups, but to a different levels of, if you like, intensity of color. But this is something we do find a little bit. So in the classic immune activation pathway, which are around the bottom of you know, this kind of these two, or the series of pink and red dot or square here in the middle, you can see that both groups are activated, possibly sometimes with a clear evidence of a kind of dose response. However, you can also see something specific about the group with CRP above 3, and in particular, an activation of kind of key metabolic pathways, such as kind of mitochondrial dysfunction and oxidative stress, which is a little bit further down. So there is something actually different from the group with CRP above 3 milligram in terms of the molecular activation at the cells level. They maybe justify why they respond to anti-inflammatory and not the others. We're just at the beginning of this data, and I think, of course, Brenda will be able to elaborate more on the importance of metabolic abnormality on top of the immune abnormality, and why this could be really actually the key to understand what is special about this group. We also found some interesting kind of findings, again, in the people with the CRP less than 1 milligram liter. So these are the ones, as I said before, that even if they don't have any peripheral inflammation with using clinically relevant biomarkers, they do present a molecular activation of immune genes compared to controls. And also we found this specific signature of a cell cycle related pathway, inhibition cell cycle related pathway. A cell cycle related pathway promotes cellular proliferation. This inhibition could be the reason why, or one of the reasons why, these patients do not get inflamed. So in other words, yes, they do have molecular activation driving the immune system, but they also have a molecular signature that keeps cellular proliferation down. And these two things compensate, and perhaps that's the protective factor. So maybe that's the reason why they are not inflamed in the first place. So we're trying to put all of this together in what we hope will you know, be a way of addressing this. And so I'm just going to introduce you to this audience a study that we just started. It's called Aspire. Here we can see our logo. There are many, I'll show you the picture, there are many principal investigators in this room in Aspire, where we want exactly try to find a way of putting all these biomarkers together and stratify patients with depression to find the best anti-inflammatory treatment, or how anti-inflammatory treatment can be used. There are multiple kind of work packages. What I want to emphasize is the kind of early work on existing meta-analysis or existing samples, biological sample from existing clinical trials, all developed going into generating a decision tool and an algorithm that would be tested into a clinical trial. Also want to emphasize that we have a very, very, very strong component of people with lived experience input into this right from the beginning. And which is, of course, something, as I said, that in this context is particularly important because of how the patient community recognize and embrace these models. And here you can see many people, Livia, obviously myself, Femke and Julia, and Brenda is here, and Andy is here, and Marion, of course, and Fondi Laura is one of the patient representative, and Courtney is here as well. So, you know, it's just by chance really that I'm so happy to announce that the study is finally starting. We got the letter from the welcome last week at the time that we had this meeting, and thank you very much for your attention.